Hi, so like I mentioned, I'm a senior. We all are. Here we are, learning about our play project and the different student topics. So I wanted to look at playfulness specifically in undergraduate research within the field of synthetic biology. So just giving some background, what is synthetic biology? Synthetic biology is an interdisciplinary field that is composed of different disciplines such as chemistry, biochemistry, biophysics, engineering, computer science, mathematic modeling, and it kind of brought to mind like, oh, the academic field that we're navigating on a daily basis is like we're coming together and collaborating and being able to hear each other's ideas to really start to solve problems. And this field really emerged quite recently in the early 2000s in MIT and Harvard in an MIT kind of like J-term class where a professor wanted to bring some students together to kind of put ideas together of how we can use the different molecular bio substrates that they use in the lab every day. And some of the different techniques that they use that define this field include DNA origami, which is kind of like Legos that build up on each other, and genome alteration, so genetically modifying things is considered synthetic biology, and protein synthesis to produce like pragmatic or entertaining creative functions and mechanisms. So like the projects sort of range from being able to create like a cancer drug delivery device to being able to do really fun stuff and make really nice structures. So my research question was, how do undergraduate students play with these biological substrates in order to learn, synthesize research projects, and shape an interdepartmental field of science? And a lot of that has to do with the actual substrates themselves because they're public domain. A lot of the knowledge is being passed on through databases, so students are able to take them from a database and have all the information they have ready to go to be able to design these things right away without having to figure out the small details. And in our play project, we look a lot at children and the way that play is something that's specific to children. So Winnicott, he's a psychoanalyst, and he's using play to understand like group relationships and how this contributes to growth. So he defines play as something that's preoccupying entertainment. It's something that you're in the zone, you're kind of blocking out the outside world. It's a form of escapism, as Lucy and Katie were talking about. And it's a way that kind of like practice your problem solving and practicality comes secondary. And there's also the familiarity of the object. It involves like moving your body in order to interact with it, and also being able to cause the response like in your interactive art. And there's a bodily response that comes with like manipulating the object to the way that you wanted it to. There he is, a handsome man. So Winnicott, I wanted to bring this back to synthetic biology. So he's looking at these group relationships, and I wanted to look at the way that interdepartmental relationships are built across like from individuals into research teams into whole fields of community of scientists, and the way that academia is able to like have this happen. And for him, play objects can be anything. It could be a kid sitting on your lap chewing on your knuckle. They could be playing with a string. They could be playing with a ball. But for synthetic biologists, they're at a nano level. You have things like DNA that build up our genetic information, but they're also used as structural devices. You have signal carriers, and you have systems that are much like circuits, like you might see in engineering, and also proteins, which are important to all of our daily functions. So here's some examples. So the first one I wanted to look at was DNA origami, and basically what's happening is it's like a string of molecules, and they're building up with each other, so you have the main structure, and then you have staple strands that are able to keep it in place. So here's a computer model up here in the rainbow. This is a computer model, and then on the bottom here is what they actually found to be what was produced after they had produced it in a thermocycling machine. So it's closely following what their models was. So here you can make anything from just a sheet to a star shape to even a smiley face. So you can do all these really nice things and make useful structures or just like beautiful art. And that's like the different scales. So it's like magnified up top and then less of a magnification on the bottom. The second thing I wanted to look at was how these systems are being built. So here we have a synthetic oscillatory network of transcriptional regulators. So I just said a whole bunch of long, big words. What this basically is just a way to use E. coli, which is a model organism that you're able to manipulate easily because its genome is really well understood, and you're able to insert a gene into it that makes it glow once it gives it a signal. But what they're doing is they're also adding a repressor layer, meaning that it stops glowing. So you have something that goes, I'm glowing, I'm glowing, I'm glowing, and then it's really bright, and then it comes back down and dims down. And it's just a cycle of bacteria colonies that are doing this. So one might ask, like, what's coming out of all this research? Like, what are people really applying these biological substrates to? And like, how are they playing with these objects and thinking about things in order to do these projects? So there's like different fields it's affecting, including the pharmaceutical industry, agricultural genomes, and biofuels are obviously really important right now for thinking about like global warming. And society's relationship to the ethics of synthetic biology is changing because you have things like, things are coming in that are like, oh, people are starting to make test tube babies, or people are starting to implant genes. But overall, because this is in an academic field and it's controlled by academics, it's limited to what students want to pursue and what they find is most useful. And it's really being shaped by all of this as a liberal arts education with collaboration. And there's a huge amount of potential for the progress of this technology to really impact this community. So I wanted to show how synthetic biology was unique, and it's because of this foundational organization of the community that allows it to be sort of playful and allows students to really engage with the material in a low-stress, non-capitalistic way. So they have these things called the iGEM and Biomod student competitions, and they're like student science fairs, like on steroids. So people get together and it's kind of like a NASCAR race, and everyone's in a team and they're wearing team, and they're presenting their projects in a way that they're able to share their information without pressure of like, oh, I'm first place, second place, or third place. They're just sharing it for the sake of sharing it and engaging with each other's ideas. And like I mentioned before, these are all open source shared biomaterials that are usually not funded by corporations, but rather by government organizations or academic institutions. So one might ask, is this academic, where it has a purpose, or is it extracurricular? Because if it's extracurricular and it's just students getting together to do it, then it's something that might seem more like leisure and being able to really play versus having it be a resume builder, which seems propulsive towards work rather than play. And I argue that it is play because there's a lot of these creative projects that are designed by students, including E. coli that smell like bananas. So if you never worked with E. coli, they smell horrible. They smell awful. But these students were able to knock out the gene that makes it smell so bad and insert a gene that allows it to make this phenolic compound that smells just like bananas. And I quite like the smell of bananas, so this is great. And another thing would be DNA origami, which is geometric, and being able to put these together as like units, but then also make larger structures from it, like you might see in a Lego kit for kids. So here, this is from the University of Sao Paulo in Brazil, and this is a project from 2015, and they were taking these nanotubes and making them into these different structures. So here you have like interchangeable parts that are able to link together and form these nano cages or even a plane structure, which can be used as like delivery service devices within a cell. And here's another project. This is from the Rutgers Biomod project. So one of the student competitions. So these are all mostly undergrads and some assistants from graduate students. And this is really important because they wanted to like see the way that these enzymes link together and are able to form these structures on their own in order to figure out how to biodegrade all these chemicals. And it's really nice geometrically looking because they have all these like beautiful fractal patterns that make fan structures. But it's analogous to almost like a childhood railroad track where you would kind of link these like flexible elements together in order to form circuitry that can actually be useful in different biomedical applications. So here's an example of the team, and they're not affiliated with biotech industries or pharmaceuticals quite yet. And this is what it was at 2012. The competition has grown quite considerably in the last 4 years, and this is at the Weiss Institute. So these are all the participants in the different teams. And as you can see, they're just like us. So what is the direction of the field of synthetic biology, and how does play and creativity motivate these participants? Well, I'd say it's more play as an aspect towards research. It's something that's grappling students' attention, wants them to be able to read the information and try to figure out how to use these different parts that are made available to them in databases that are resume builder. And a lot of it's pleasure because it's learning, and it's being able to develop these skills without having the pressure to apply it for a patent later on. And some of the project topics include like what specific components of the research projects can be seen as a form of play, and how can this research be moved into the classroom. And moving forward, with play as a form of learning and motivation, and synthetic biology being conducive to this creative process, I wanted to look at the collaborations across disciplines for developing problem-solving skills, and how like synthetic biology engineering organisms could produce biofuels more efficiently. How are students being able to apply the materials that are easily made available to them and the information in order to like learn these processes of like, okay, this is like how photosynthesis works, this is how like we're using biofuels, and this is how we can apply it to society. Yeah, so, thank you.